Welcome back to our Jepson mini workshop on biodiversity challenges, California floristics, taxonomy, and phylogenetics. And this will be module 3, which is examples of phylogeny-based revisions. So I talked earlier about monophyletic groups or clades, which represent all the descendants of a common ancestor. And these are similar examples to what I showed earlier. And a point I just want to make in further discussing monophyly or clades is that all members of a clade share the same level of relationship with members of another clade. So for example, these 3 terminal taxa on this leftmost tree shown here are each equally closely related to the lineage just outside that group. So it could be that there have been different rates of evolutionary change along these 3 different branches, or maybe along this branch, that could mean that these individuals are not as similar or equally similar to the individual out here in terms of overall characteristics. But in terms of recency of common ancestry, each of these 3 members of this designated clade here share the same most recent common ancestor with members of this group. So we're always looking down to these divergence points and thinking about recency of common ancestry as measure of relationship. And I bring this up in part because there is another common outcome when one looks at relationships among members of traditional genera or families, for example, and that is that those often turn out to be what we call paraphyletic rather than monophyletic. So a paraphyletic group is also called a grade rather than a clade. And it's basically defined as including some but not all the descendants of a common ancestor. And each of these 5 examples shown here are different grades where we have the different members of this group in each case are united by common ancestry, and that is the grouping criterion that resulted in taxonomists proposing such a group, very likely based on homology, that is similarity due to common descent. But some members of the grade are more closely related to lineages outside the grade than to other members of the same grade. So here, for example, these 2 species are terminal taxa at the end of this particular clade here are more closely related to this lineage that's not part of the grade than they are to these other lineages within the grade, which have a more distant common ancestry than these 2 do at this point. And so this situation commonly arises in cases where we've had an acceleration of evolution along one of the branches, or potentially a deceleration within the grade, such that overall similarity is misleading about relationships. And it's important to recognize such problems if we want our taxonomy to be based on recency of common ancestry, and to be able to use the data for comparative biology, the taxonomy, to reflect groups that should be subjects of comparative studies and for conservation biology. And I just want to point out some examples recently, or fairly recently, within the California flora where we've seen these kinds of changes where a paraphyletic taxon has been revised to render it monophyletic. And so this is a case regarding the shooting stars in the primrose family, which if you've been involved in California botany for a while, you probably remember when they were considered part of the genus Dodecatheon. And here's a representative shooting star here, very distinctive morphology of the flowers. Whereas it turns out that here we see Dodecatheon in this tree. Basically, everything to the right of this branch here is Dodecatheon in its traditional sense. You can see it's a monophyletic group. Dodecatheon represents all the descendants of a common ancestor at this point in the tree. This is a molecular phylogeny produced by Austin Mast and colleagues. But we can also see that Dodecatheon is nested phylogenetically within a grade called Primula. So here we can see these are all different species of Primula forming lineages branching off of this backbone here. So if we look at the base of Primula, which is actually off the bottom of this slide, it would not only include all the species of Primula, but also all the species of Dodecatheon. So Primula is paraphyletic without including the shooting stars. Now it's important to realize that this phylogenetic result was super informative, because this pattern of having one genus nested in within another basically suggests that we are dealing with a grade with a nested clade. And in that kind of a situation, you're generally seeing a lot of change along the branch that's at the base of the nested group, in this case Dodecatheon, where there's at least 6 major trait changes that have been mapped there, including the reflection of the petals and the fusion of the stamens to one another, their enlargement and production of a lot of pollen, the opening of the anthers through a pore initially before longitudinal dehiscence. All of these traits associated with buzz pollination by bees, which is a transition that happened at this stage in the evolutionary history of the primroses, where we went from a flower like this, like we see here, to a shooting star flower associated with a shift to buzz pollination. So this kind of shift in pollination syndrome can be associated with major divergence events. And this is a great case in point. But you can see here that the closest relative of Dodecatheon is our only species of non-shooting star Primula in California, Primula suffrutescens here on the left, that has similar coloration to its flowers that appears to be a shared derived trait. And there are some traits that have changed along the backbone of Primula that lead up to this point where the buzz pollination characteristics started to evolve, that sort of set the stage for buzz pollination here. So evolutionarily, a really informative tree, but in terms of how to deal with this taxonomically, kind of a conundrum. So there are a couple of alternative solutions to paraphyletic taxa. One is to include the nested taxon, in this case Dodecatheon, within the paraphyletic taxon, as in Primula. And then we just have to choose whichever name is oldest, and it happens to be Primula for that larger taxon. And this is the option that Austin Mast and Jim Reveal took in resolving this problem, wanting the generic taxonomy to reflect the close relationship between the shooting stars and other primulas. But another alternative solution would be to recognize segregate genera of the paraphyletic taxon, that is to break up Primula into a set of monophyletic genera, which in this case would require several of narrow delimitation. And also the question arises, are these segregate genera readily diagnosable, for example, morphologically? So there can be some taxonomic usability criteria that come into play, whether to choose that particular option. So deciding between these alternative taxonomies then really involves considering the relative gain in information content of one versus the other, and the level of disruption to taxonomic stability of choosing one approach to revising the taxonomy versus the other. And that's a really important criterion. So we definitely want a taxonomy that's usable and where the major groups are diagnosable, hopefully morphologically, but we also don't want to disrupt the stability of taxonomy unnecessarily, because taxonomy really serves an important function in communication about plant diversity, as everyone can appreciate. And if the names are constantly changing, it has a disruptive effect that hinders communication, which everyone agrees is not great and it should be minimized. We still want our taxonomy to be evolutionarily informative, but we want to make those changes in ways that are least disruptive to stability. A similar solution was put in place for the snapdragons when it became clear that Mohavea, a genus of 2 species, both in California, down in the deserts, is nested highly within Antirrhinum, the snapdragon genus. And here we can see a phylogeny of Antirrhinum, again, tilted on its side, so the names of the species are out here on the right. And here we can see Mohavea, a clade again, as was Dodecatheon, but again, nested within Antirrhinum. And as it turns out, the Antirrhinum convertiflorum, the ghost flower, was originally treated as a species of Antirrhinum. The name was already available in Antirrhinum for that plant. And it's quite readily accommodated there. There are some interesting floral changes that have happened in Mohavea, but it can be accommodated morphologically within Antirrhinum in a way that doesn't make the genus too heterogeneous to be diagnosable. And it's less disruptive to the stability to change 2 species names to Antirrhinum, one name already available in the case of convertiflora, than to break up Antirrhinum into a bunch of different groups, and relationships in Antirrhinum a bit unstable anyways to make that difficult. So the option that Dean Taylor and Dave Keel chose was to submerge Mohavea within Antirrhinum. But I should point out there are other botanists that have gone ahead with the breakup of Antirrhinum instead. So alternative taxonomies are possible, both based on monophyletic groups, but it's a question of how much you value taxonomic stability and morphological diagnosability, and how much you trust the phylogenetic data, for example, in terms of what you decide to do. But the Jepson Flora project has gone along with the enlarging of Antirrhinum to include Mohavea. A different solution, though, was instituted for Chlorogalum. Chlorogalum, the soap plants, are members of the subfamily Chlorogaloideae, which is a subfamily of the Agavaceae, the agave family. And phylogenetic studies using molecular data, DNA sequence data, by Halpin and Fishbein, first showed that Chlorogalum is not monophyletic, and that in fact the night flowering species of Chlorogalum constitute a well-supported clade or monophyletic group, but they're more closely related to members of the genera Hastingsia and Camassia, the camas genus, than they are to the day flowering species of Chlorogalum. And here we can see a night flowering species of Chlorogalum, Chlorogalum pomeridianum, very common member of our flora, and one of the members of the day flowering Chlorogala. This is Chlorogalum purpureum. And in this tree, it looks like the 2 day flowering species of Chlorogalum themselves form a paraphyletic group, bracketing off like this. But a subsequent study by Archibald et al. With more data showed that these day flowering species constitute a clade. And so the decision that was instituted, involving again one of our Jepson editors, Dave Keel, was to erect a new genus, Hooveria, named after Robert Hoover, who had strong feelings about the distinctiveness of these day flowering species of Chlorogalum, for the day flowering species, a new genus for these. And so this was also the least taxonomically disruptive solution, and the solution that resulted in a taxonomy, a generic taxonomy that was very nicely diagnosable. Expanding Chlorogalum to include all of these things like camas and Hastingsia was pretty morphologically heterogeneous. So that solution wasn't taken. And the last type of the 3 major types of relationship, monophyly, paraphyly, the third type is polyphyly. So a polyphyletic group represent the descendants of 2 or more disparate ancestors. So we see different examples here of polyphyletic groups. that are not united by common ancestry. So it's a grouping criterion based on false homology. That is, the similarity that led to these plants being treated as the same within the same taxon is due to independent evolution of the characteristic. So it could be parallel evolution, convergent evolution, reversal evolutionarily. And such a grouping is wholly artificial based on from an evolutionary standpoint. And everyone agrees that polyphyletic groups are not to be recognized if you have any kind of an evolutionary criterion with regard to taxonomy. And a good example of a polyphyletic group that's been recently revised are the hounds-tongues, the genus Cynoglossum. And the way that it had been circumscribed, it included representatives of several major lineages of the Boraginaceae. As you can see, these colored groups here in red here. So there's several different groups representing Cynoglossum. So the traits that had been traditionally used to lump these plants together in a common genus independently evolved. These broad leaves and colorful flowers. And our New World hounds-tongues, the type of Cynoglossum is in the Old World. Our New World hounds-tongues are now in the genera Adelinea and Anderson-glossum, which actually are more closely related to things like Cryptantha and Amsynchia, Plagiobotheres than they are to any of the species that they were traditionally placed with in Cynoglossum. So this was an important change in taxonomy. An extremely artificial group as traditionally circumscribed. Another example are the broom rapes. So Orobanche, as it was historically circumscribed, all the members of it were definitely members fall within the same large clade that's been called the broom rape family, Orobanchaceae. But if we look at the relationships within Orobanchaceae, we see that the Old World Orobanches form a clade that is not most closely related to the New World Orobanches down here, which are more closely related to the Old World genus Phylogeny. And there's quite long branch lengths here. This was well-supported result. And so the Asa Gray genus, Aphyllon, which hadn't been recognized in many, many years, was revived as an available name at the genus rank for this clade of New World, all the New World broom rapes that had been in Orobanche before. And we'll get back to this group later. Also in the Themidaceae, the blue dicks have become blue dips. Dicolostema capitatum is not as closely related to the rest of what had been treated as Dicolostema, which includes the type species, the rest of the genus, as the core of Dicolostema is to Brodea. And these core Dicolostema species have 3 fertile stamens, as does Brodea in its narrow sense. You can see a clade here constituted by Brodea and Dicolostema to the exclusion of blue dicks, Dicolostema capitatum, which has only 3 fertile stamens. Which has 6 fertile stamens with the outer stamens reduced. But it comes from an ancestral condition of having 6 fertile stamens. And the conversion of 3 of those fertile stamens to staminodes is one of the morphological markers of the clade that unites Dicolostema in the new sense, new constricted sense, and Brodea. So in this case, there was definitely a morphological hint that there was a problem. And now we treat blue dicks as Dipterostemon, which is an old name revived for them, and their common name is changed accordingly to blue dips. Okay, so those are some examples of changes involving paraphyletic and polyphyletic groups. And now I'll open it up to questions.